Every second, mitochondria, the microscopic powerhouses of our cells, fuse, break apart, and reorganize. They manage energy, detoxify waste, and shape how cells behave. But what if these tiny structures did more than power life? What if they helped decide what kind of life a cell becomes? That's exactly what Dr. Chun Ju Chang and her collaborators proposed in a 2019 study published in Cell Metabolism. Their findings revealed a direct link between mitochondrial dynamics and the identity of stem cells, offering a new lens into cancer progression and regeneration. Chang's journey to this discovery spans decades and continents. Born and raised in Taiwan, she later completed her PhD at UCLA and postdoctoral training at the University of Texas MD Anderson Cancer Center. With stops at Roswell Park in New York and now a professorship at China Medical University in Taiwan, she has pursued questions at the intersection of cancer biology, cell signaling, and drug discovery. Across these roles, she has received over a dozen recognitions, including the Angin Award in Basic Science Research and the Women in Cancer Research Scholar Award. But her most meaningful contributions often appear in the achievements of her students. The 2019 study focuses on a cellular process called epithelial mesenchymal transition, EMT. EMT is known for its role in cancer metastasis, allowing cells to detach, move, and invade new tissues. But EMT also grants cells some stem-like traits, which can make tumors harder to treat. Chang and her team found that EMT causes mitochondria to fuse, forming long, tubular structures rather than fragmented ones. This change is not just structural. It increases antioxidant capacity and promotes self-renewal in mammary stem cells. The fused mitochondria, they discovered, are directed into at least one daughter cell during stem cell division, helping preserve a pool of renewing stem cell population. At the heart of this process is MFN1, mitofusan-1, a protein required for mitochondrial fusion. The study showed that MFN1 doesn't just regulate mitochondrial shape. It also interacts with polarity proteins like PKC zeta and NUM, guiding how the fused mitochondria are split during cell division. This finding has significant implications. Cancer stem cells are thought to drive recurrence and resistance. If fused mitochondria help sustain those cells, then targeting mitochondrial fusion, particularly MFN1, could be a way to make tumors more treatable. More broadly, the study challenges assumptions about what drives stemness in cancer. While transcription factors and signaling pathways are well-known players, Chang's work shows that the physical state of organelles also plays a critical role. Her team even demonstrated the effect in vivo using genetically modified mice. Deleting MER200C, a microRNA that limits MFN1, caused excessive mitochondrial fusion and an increase in stem cells in the mammary tissue, linking EMT, mitochondrial behavior, and stem cell expansion at the tissue level. Despite the complexity of her findings, Chang is passionate about making science approachable. As a professor at China Medical University, she emphasizes hands-on learning and encourages students to challenge assumptions. In class, she breaks down advanced concepts in cell signaling and metabolism with visual models and analogies. In the lab, she helps students design clean, logical experiments, often challenging them to predict outcomes before touching a pipette. Chang credits her own mentors at UCLA and MD Anderson for showing her the value of rigorous but supportive training. Now, she strives to pass that on, especially to young women entering cancer research. She advocates for greater representation in leadership and speaks often about the structural barriers women face in academia. Today, Chang continues to investigate how mitochondrial dynamics affect cell fate and how these insights might be leveraged in therapy. Her recent work explores whether disrupting MFN1 interactions could selectively deplete cancer stem cells, or whether boosting mitochondrial control might improve regenerative medicine. She's not done asking questions.